In this video, I'm going to walk you through the rest of the organelles and set the stage for us to understand how things can and can't get through the plasma membrane. Remember the cytoskeleton? Do you remember which organelle is the puppet master using these little cytoskeleton fibers to move things around? It's, of course, the centrosome, shown here in red. And we see here that the cytoskeleton has a number of different types of little fibers. Some of them are little tubes called microtubules. Some of them are little filaments made of a protein called actin. Actin like action. This allows movement. Very interesting. This is another of the muscle proteins too. We'll talk about that and how your biceps, for example, is capable of shortening. There's actin in muscle cells in abundance. Intermediate filaments give us some mechanical strength, and then there are some little lipid droplets and melanin pigments sometimes that start to stick to those cytoskeleton components. Let's look a bit more closely at the nucleus and some of the other organelles. Remember RNA? RNA is actually 4 chemicals, and we use these letters to represent the 4 chemicals, G, A, U, and C. Do you remember that from a prior biology class? DNA has 4 chemical building blocks too, and 3 of them are the same, G's, A's, and C's, but there are no U's in DNA. There's a T instead. Do you remember that? GATC, GAUC. That's just a quick preview. We'll go into a lot more detail talking about those in a little bit. The other main part we need to know about the nucleus is that it has bound ribosomes for synthesizing proteins, which can pass into the nucleus, things like transcription factors and enzymes. And there are nuclear pores, these larger ones here. They allow chemicals to come out and in, electrolytes and energy in the form of ATP. Whoops. When we talk about DNA, of course, this is the blueprint for life. These are the 4 chemicals I was talking about. G, A, T, C. 4 chemicals, like 4 letters, can be combined to spell out different words. In the same way, these 4 genetic so-called nucleotides can be combined to spell out the recipes for thousands of different proteins. And your unique physical body and a lot of your personality traits actually have everything to do with your genes, the letters and the order of those letters spelling out your unique recipe. That DNA comes in 2 different forms. Sometimes we want it all unwound as one long noodle, like a spaghetti pile. In this way, we can read the recipe in the genes, and we can convert that recipe into a portable document through a process called transcription. That is the first important step in protein synthesis, this gene expression. Sometimes, primarily in mitosis, when we want to divide the cell, we have to wind up the spaghetti around these little histone proteins and then wind those together until the long spaghetti noodle is in a nice compact package, sort of a X-shape chromosome, right? So we have chromatin and chromosomes, and if you look closely, you'll see that this DNA is actually identical to this DNA, having already been copied. This chromatid is an identical copy of this chromatid, and together the 2 of them are called chromosomes. We talked a little bit about the puppet masters before, the centrioles, who in that pair are called a centrosome, and the fact that they pull on these little tubes and filaments. You can push them too, by the way. Ribosomes. Ribosomes are the site of protein synthesis. We'll see that in our gene expression video. They actually read the RNA code and then begin to put amino acids in just the right order. Everything you need to know about the endoplasmic reticulum right here. Some people call the Golgi the shipping department or the post office. Anything that was made in the cell, most things anyway, have to go through the shipping department to be properly packaged and labeled for export. In a more scientific description, the Golgi has additional enzymes in it which help to tweak the shape of the protein. So the primary protein coming in the transport vesicle might be close, but not exactly the right shape. And the further manipulation and finishing adds or breaks certain hydrogen bonds to fold the protein into exactly the right shape, where it is then packaged for export. This is exocytosis. I talked about this in the preview, but remember, if some viruses or microbes or garbage end up being formed into a vesicle, this inward drinking or eating of that material is called endocytosis. And we have a vesicle here, packaged in a membrane. Because the lysosome, the cleaning agent, is made of the same phospholipid bilayer as the vesicle, they can bump into each other and fuse. And then we have like a whole lot of fruit ninja going on in here. The enzymes are chopping up all of those little germs. Remember that mitochondria are actually foreign organisms living inside of you. How amazing. We'll have the opportunity to talk about that a little bit more in class. Here's really what I need you to know. Mitochondria are capable of taking the tiny particles from our food in these forms. Carbs are ultimately digested into glucose, and fats, avocado and bacon and things like that, are ultimately broken down into fatty acids. And proteins, like this turkey, or tofu, or beans, are digested into amino acids. And look at what the mitochondria can do. It can convert these through additional steps not shown here into a chemical called pyruvate that goes inside, and we can completely oxidize that pyruvate. And we're left with a toxic byproduct called carbon dioxide. What do we do with the chemical energy, the so-called calories in the food that we eat? We harvest the chemical energy by breaking or oxidizing that food, and we store that energy in this form of ATP, the rechargeable battery, which is then used to do work. I mean this in the physics sense, right? Work. Sometimes that means movement. Sometimes that means cellular transport. Sometimes that means generating an electrical voltage. And the byproduct of all of that biochemistry is heat, which is why we are warm-blooded creatures, right? It's advantageous, but we're really just super inefficient engines. We're wasting most of our calories as heat. Great time to pause the video and reflect on all the things you've learned in the first 2 modules. Can you identify all of these parts of the cell? Can you describe their function? Can you make an unlabeled version of this picture to study at home? I'll show you how. Watch this. Duplicate the slide. Cut the picture out. Let's see. Cut the picture out. Oh, you know what? That's not going to work. If the labels are of a different image, then you can cut it out. Here, I'll show you what you can do with this one, though. You can crop out the words, if you so choose, to make an unlabeled one. When they put the labels on the periphery, it's a bit easier to do. Otherwise, if you go into an image, oftentimes the labels are additional little graphics, and so you can just cut out the picture. But look at that. Now you can really test yourself. Can you identify all of the parts of the cell? Good luck.